Thank you, Adam. So now I'm going to look at the projects that NHGRI funds under functional element discovery, characterization, and interpretation. Our largest project in this area is the ENCODE project, which is currently funded through the end of calendar year 2020, actually 1 month into 2021. ENCODE has already been mentioned but I just want to quickly go over the main goals, which are to map candidate functional elements in the human and mouse genomes. This is by mapping a number of different types of biochemical activities, including transcription, open chromatin, histone modifications. They also have an ongoing activity to functionally characterize these candidate elements in specific biological contexts, using a wide range of technologies, some relatively low throughput, some very high throughput, and I think we're going to hear about more of these tomorrow. Main goal for ENCODE is to create a freely accessible data resource. And this includes data that's been collected using unrestricted data sharing consents that allow for unrestricted data sharing, that is. ENCODE is compiling an encyclopedia of regulatory elements. In addition, there is a set of grants looking to develop computational tools to maximize our understanding the value of ENCODE data. And ENCODE shares uniformly processed data as well as processing pipelines on the cloud. To date, there's about almost 15,000 data sets on the ENCODE portal here under encodeproject.org. You can find that. And about 10,000 of those data sets are from ENCODE, and the remainder are from associated projects such as the Roadmap Epigenome Center activities, as well as fly and worm data. So a lot of what we've been talking about are named projects, but I want to point out that NHGRI does support quite a bit of investigator-initiated research. I don't have the exact numbers, but somewhere close to half of the extramural budget is actually focused on investigator-initiated activities, depending on how you categorize them. And so in particular in this area, we've recognized recently the real importance of trying to connect from relating genetic variation to function and disease. And we recently put out a program announcement to support the development of novel and generalizable approaches for how genetic variation leads to differences in function and how such functional differences affect human health and disease processes, or how this knowledge can be used clinically. And so I just want to emphasize here, we still have quite a lot of interest in investigator-initiated research, and hope that that will be part of the discussion here today. I'm just going to highlight 4 other related projects that are funded by NIH, as well as the last being a more international consortium. There are people in this room who know a lot more about these than I do, and so if they have questions, we can call on them. But the first is the 4D Nucleon Project. This is a common fund project to understand the principles behind the organization of the nucleus in space and time. Also, they understand the role of nuclear organization that it plays in gene expression and cellular function, and how changes in nuclear organization affect normal human development and disease. The second is another common fund project called COMP2 for a knockout mouse phenotyping program, and this provides broad standardized phenotyping of a genome-wide collection of mouse knockouts. And then we have 2 more cell atlases that kind of build on what was talked about earlier today. The first is another NIH Common Fund activity for the Human Biomolecular Atlas Program, or HUBMAP. And this facilitates research on single cells within tissues by supporting data generation and technology development to explore relationships between cellular organization and function, and variability in normal tissue organization at the level of individual cells. And then there's the Human Cell Atlas, which is an international based research consortium with the goal of creating a collection of molecular maps of different cell types where they are located in the body, with the goal of enabling our understanding of the cellular basis of health and disease. And so I just want to summarize what Adam and I were just talking about, that this is a good time to take a fresh look at NHGRI's programs in this area. As the landscape is changing, we've provided this summary for you, but as Adam mentioned, it is probably not comprehensive. It gives you a good idea of other activities. Many of these are taking place outside of NHGRI, and the question is how can we best integrate across this, help and complement those activities. As was mentioned a couple of times, the major NHGRI programs in this area, genome sequencing and ENCODE, are coming to an end of their current funding in the next year, 2 years. And so we want to hear about the opportunities, particularly at the intersection of these two major areas. And with that, I think we're going to move into the more detailed discussions, scientific presentations, and I'll turn it back over to Adam.
Yeah, at this point, I'd like to invite the state-of-the-art speakers up. Those will be Amit Kara, Laura Beirut, and Jonathan Haynes. You can all come up and sit, or you can come up one at a time.